Jennifer Kneister. I'm a senior at the University of Michigan, and I'm the project lead for the Michigan Synthetic Biology team. This year, our project is called Aptapaper. Aptapaper is a paper-based gene network that is designed to detect proteins secreted by a pathogen in order to detect diseases. So, in a recent Cell publication, it was demonstrated that transcription and translation could occur on paper. So what they did is they took these DNA circuits, freeze-dried them on paper, then you add this in cell-free expression kit that translates the DNA into an RNA switch, and then you can induce that switch with a piece of RNA. So then your switch can have like whatever protein output you want. Generally, it's going to be a visual reporter protein. So to make use of this, I thought it would be best to detect a protein because proteins are secreted by pathogens. So then you can use it to detect disease. So in order to hook up the protein to the DNA switch, we're going to use something called a DNA aptamer, which is just a piece of DNA that folds in a special conformation so that it binds tightly to a specific protein. So aptamers can be designed for whatever protein or molecule you want. So you can easily make an aptamer for a different protein. So basically, we're going to take this aptamer and then this gene circuit, and we're going to hook them up together and use that to detect a disease so that when you have your pathogen-secreted protein, you get a visual output. And that all occurs on paper. So this is what our system would look like without the presence of a protein. You have your DNA aptamer and your trigger for the switch bound together. So that way your trigger can't go and trigger your switch. And then the idea is that when your protein comes in, it binds to the aptamer, and it binds tighter to the aptamer than the piece of DNA binds to the aptamer. So that forces this piece of DNA to displace, and then that can go trigger the switch. So this is kind of a special type of switch where it has something called a toehold region. Basically, the toehold region is outside of the stem loop, so it's really easy for your trigger to come in and bind. So that's the same region that's bound right here. So that way it makes it harder for the switch to accidentally be triggered. So this toehold part of the thing is going to come in here, bind here. That'll unzip the rest of the switch. That exposes your ribosomal binding site. There's your AUG start codon, and your reporter can be expressed. Okay, now I'm just going to draw this same thing, but what happens when you have a protein present. Here's a quick picture of what the system looks like when your protein is present. So your protein binds tighter to the DNA aptamer than your trigger does. So your trigger is displaced and toehold region is exposed, so it can come in here and start unzipping the switch, and then your ribosomal binding site is exposed and your reporter can be expressed. So why should you care about this system? Well, currently there's a need for improved disease detection, in particular for tropical diseases. Some diseases affect not a great number of people, but then there's a lot of diseases, so it adds up to be a lot of people. These diseases tend to be in remote areas where the current tests require a lab. They tend to be expensive. So basically, they're not fast, they're not cheap, and they can't be done on site. So that's where our paper-based gene circuit idea comes into play. Basically, you have this system, it's freeze-dried to a piece of paper, and all you're going to do is put a drop of somebody's blood on the paper, and if they have the protein that indicates the disease, then you have a visual output, and you can see that response within a couple hours. You don't need any additional equipment at all. To continue working on our project, we need your help. Some of the kits and reagents we're using are very expensive, even though our final product is predicted to cost cents per device. With your help, we can take our project to the iGEM 2015 competition. You can expect frequent updates on our work throughout the summer. Thank you so much for watching, and thank you very much for your support.